So hello everyone. I hope we are doing well. So welcome to our video today. Today we'll be doing a short introduction to embryology. We've done introduction to gross anatomy, introduction to histology, and today we'll be doing introduction to embryology. So we'll start us off by looking at some of definitions that we'll include in today's discussion. And the first one is embryology itself. So if you like to define embryology, we'll define it as the development of an organism from the time you'll have fertilization to birth. So fertilization is when the male and the female gametes fuse to form a zygote, and then birth, of course, is during the delivery period. So embryology is that period, the development of now that zygote until it becomes um, a baby. Then for basic embryology now, remember we'll divide embryology into two, basic embryology and systemic embryology. For basic embryology, these are now the general stages and process of development of this embryology. So the general stages of fertilization, the general stages of implantation, those are basic embryology and they are known as basic embryology. So the general stages and processes of development of this embryo are known as basic embryology. Then for systemic embryology, these are development of individual organ systems. So for example, if you want to develop the skeleton, the exoskeleton, or maybe the skin, the integumentary system, you'll study that system by its own. So that systemic study of embryology is known as systemic embryology, which is a development of individual organ systems. Of course, we have a couple of organ systems that we'll see in our later videos. For comparative anatomy, of course, it has the word comparative, which is compare. So you're trying to compare the comparison of embryonic development of various species to understand evolution. So you can study the development of human embryo vis-a-vis the development of the embryo of a monkey. So that is comparative anatomy or primatology. So comparative anatomy, it's a branch of primatology in it, but it's under embryology. So comparative anatomy, you are comparing embryonic development of various species to understand our evolution. And the last definition of the day is teratology. Of course, we know teratology is a studying of congenital abnormalities and now the factors that are leading to these congenital abnormalities. So which are developmental defects. So studying of congenital abnormalities, factors, the time, maybe smoking, drugs that can cause developmental defects. So some congenital abnormalities, they are occurred at a specific period of time and due to maybe predisposition to a certain factors. So those factors will be studied in a special session as teratology. So, but ideally, if you have to define teratology, you talk about as a studying of congenital abnormalities and factors that cause developmental defects. Yeah, so there are periods of development. So you'll divide it into two. Before conception, conception is fertilization, and after conception. So you'll divide this period of embryology into two: before conception, before fertilization, and after conception, after fertilization. So before fertilization, it will be this is the period um, when now the fertilization is happening, or even before the fertilization happens. So we'll have the process of gametogenesis. So gametogenesis is the process of formation of gametes, which are sperms and ova. And then also we'll have fertilization. So before conception, you'll have gametogenesis and fertilization. Of course, gametogenesis is, you'll have two, spermatogenesis in males, that is formation of sperms, and then oogenesis, formation of ovum in females. And then fertilization, of course, is the fusion of the male and the female gametes. After conception, you'll divide it into three periods. You'll have pre-embryonic period, embryonic period, and the fetal period. So pre-embryonic period is a period between conception and the first 2 weeks. So you'll have first week of development, second week of development. The embryonic period will have from the third week of development to 8th week of development. Then fetal period is from the 9th week of development up to birth. So ideally, pre-embryonic period from 0 to 2 weeks is where you have now until you have the embryo being fully implanted and the placenta is starting to form. Then embryonic period from week 3 to week 8 is now when these general processes, neurulation are happening. When that fetal period is now when you have now the organ system starting to develop. So the organ systems start to develop within the embryonic period, but now they are finished up in this fetal period. So that is how you divide the periods of development: before conception, after conception, and then you have the specific periods. We start off with gametogenesis, because gametogenesis is the process of formation and development of gametes. And these gametes are formed. These gametes refer to ovum in females and of course sperms in males, and the processes usually occur in the gonads. So gonads, there you have the testes in males and the ovary in females. So gametes, sperms, males, ovum, females, develop in gonads, which the gonads are testes in males, ovary in females. So specifically now, oogenesis is the process formation of ovum in females, while spermatogenesis is the process formation of sperms in males. So I presume that one we kind of know. So we start us off by looking at oogenesis. So for oogenesis, you can see this image is actually showing us an ovary. So the ovary has two parts. That's cortex. This is the ovarian cortex, the outer parts. Then there is a ovarian medulla. And this ovarian medulla, you can see it has ovarian ligaments with ovarian blood vessels. So oogenesis is now, which occurs in the ovarian cortex. It has various follicles which develop to become ovum, mature Graafian follicle at specific periods, so within 1 month. So stages of development, you'll have primordial follicles which undergo mitosis. So you can see these are primordial follicles. They undergo the process of mitosis. Then from primordial follicles, you'll go to primary follicles. In primary follicles, they undergo meiosis 1. Then you have secondary follicles which will undergo meiosis 2. You'll have mature Graafian follicles. Now, when you have mature Graafian follicles, they are ready for ovulation. And actually, you can see from primordial follicles, you go to primary follicles. From primary follicles, you go to secondary follicles. From secondary follicles, go to, this is actually the mature Graafian follicle. Then ovulation happens here. So the remains of Graafian follicle, the mature ovum, it will be the corpus luteum. And this corpus luteum now will develop. And now once it starts to generate, it will become corpus albicans. So that is the process of development of the ovum. We need to appreciate these are very important image. It will be there a lot in exams. So you'll be asked parts of mature graph and follicle or a mature ovum. You may be told to draw, and this is how you're supposed to draw the image. So secondary follicle, ideally, this at the center here. This is secondary oocyte, the secondary oocyte surrounded by zona pellucida. Zona pellucida is a protective layer that prevents polyspermy. Then you have this layer of cells on the outer bit here. These cells will be known as corona radiata. Then the cells on the opposite side will be known as cumulus operans. Then you'll have these six. This is follicular and the layer of fluid that is found there. Then you'll have now other cell types starting from here. So here you'll have the granulosa cells, and then 7 you'll have theca interna. On the outside bit now, maybe around this place, you'll have theca externa. So those are various layers of cells and parts of mature graphic follicle. So this is secondary oocyte, zona pellucida, corona radiata, cumulus operans, follicular antrum, granulosa cells, and theca interna cells. So kindly take note of those cell types and even the parts of that mature graphic follicle. We can go to the female menstrual cycles. Of course, it takes place in a period of 28 days. It can vary from one person to another, but it's important we know the way the various um, hormones peak. So the first hormone to peak is a follicle stimulating hormone. So this is a follicle stimulating hormone. So it peaks that can allow the follicles to develop. So remember the primary follicles, secondary follicles, they have to develop. So that's the reason why this follicle stimulating hormone, which comes from the pituitary gland, peaks. Then from there, you'll have peaking of estrogen, which is produced by ovary. So it peaks. So that when it peaks, it even stimulates this luteinizing hormone to be produced. Remember, luteinizing hormone just peaks before day 14 to stimulate the process of ovulation. So the first one to peak is follicle stimulating hormone, followed by estrogen, of which now estrogen stimulates luteinizing hormone. Luteinizing hormone stimulates ovulation. That's the reason why it's just before day 14, as you can see here. Then from there, the one that is peaking more after day 14 is progesterone. So progesterone is peaking to allow now thickening of the endometrium and in preparation for implantation and also things to do with maintaining pregnancy if fertilization happens. So those are the four important hormones that you're supposed to know: follicle stimulating hormone, estrogen, luteinizing hormone, and progesterone. So very low levels will be present in females of testosterone. Very low levels of testosterone will be present in females, as you can see there, though it's present. We go to spermatogenesis. Spermatogenesis, of course, is the process of formation of sperms. In males, it takes place in the seminiferous tubules. So what you are seeing here is actually seminiferous tubules of the testes. The supporting cells will be the Sertoli cells. You can see here we have Sertoli cells, which are the supporting cells of spermatogenesis. So stages of development, we'll start with primordial germ cells. So primordial germ cells will be around here. So these primordial germ cells will undergo differentiation to give you spermatogonia. And then this spermatogonia will undergo mitosis and will undergo first of all spermatocytogenesis, that is modification to become spermatogonia, and then they undergo mitosis. Remember, you have two types of spermatogonia. You can have spermatogonia type A and B. So mitosis is the one that now will be leading to differentiation of that spermatogonia. So from spermatogonia, you'll go to, so what you're seeing here is actually the spermatogonia. From there, you'll go to primary spermatocytes. So primary spermatocytes will undergo meiosis 1 to give you secondary spermatocytes. So secondary spermatocytes undergo meiosis 2 to become spermatids. And then these spermatids undergo spermiogenesis to become spermatozoa. So spermiogenesis is the process of modification. For example, you can see here in the spermatids, they don't have a tail, they don't have a good number of mitochondria, they don't have a chromosome. So spermiogenesis is the process of modification of the spermatids to become spermatozoa. Then when you have these spermatozoa now, which are now mature sperms, they'll undergo the process of spermiation. Spermiation is being released from the seminiferous tubules to the epididymis for storage, where they undergo capacitation, and then when they are released from epididymis, they undergo decapacitation. So that is the process of formation of spermatogenesis. So it's important that at least we know how to draw this also. From primordial germ cells, spermatogonia, primary spermatocytes, secondary spermatocytes, spermatids, and spermatozoa, and the processes that happen in between. So for example, here is mitosis, meiosis 1, meiosis 2, spermiogenesis, and then here you have the spermiation, storage, and capacitation. We're supposed to know parts of mature sperm, which is quite simple. So this one is after it has undergone now spermiation. So we start with the head, of course. A, this is a head, and you can see the head has a very large nucleus, and then it has an acrosome. The acrosome has proteolytic enzymes that enable now penetration of the zona pellucida during fertilization. Then we have this B being the neck, X is a middle piece. Then you have a lot of this tail, very large tail, as you can see there, which is now will be divided into two. We'll have like a larger part of it being the principal piece, then Z being the end piece. So acrosome, nucleus, that is a head, that is a neck, that is a middle piece, principal piece, and then you finalize with the end piece. So we cover fertilization then, as you can see there. So that's an ovum. Here's an ovum, which will be fertilized by one single sperm. If it is fertilized by maybe a multiple number of sperms, that will be polyspermy, and even it will lead to molar pregnancy. So what is fertilization? Of course, you know fertilization is a process of fusion. So the word is fusion of the sperm and the ovum. So remember, these are the male and female gametes to form a zygote. And this process occurs in the ampulla of the fallopian tube. As we can see here, we have the various parts of fertilization, but now specifically, we'll talk about the parts of the fallopian tube. We have a fimbria next to the ovary, infundibulum, the ampulla, and the isthmus. So it happens normally in the ampulla. So sometimes it can happen in the infundibulum, sometimes in the isthmus, which is still okay. So that is the process of fertilization. So fusion of male and female gametes to give you a zygote, which occurs normally in the ampulla. So what is the importance of fertilization? So why do you need fertilization to happen? So fertilization is important because it leads to formation of a zygote from the otid stage. So otid stage is just the period immediately after fertilization. Formation of zygote from the otid stage. Then we have completion of secondary oocytes, which is actually arrested at second meiotic division and formation of the second polar body. So this is the process of oogenesis being completed. So fertilization allows secondary oocyte to the arrest, which happens in the secondary oocyte, to be completed, and formation of the second polar body. Then remember the gametes are usually haploid in nature, so they will be restored to become diploid in nature. So the 2n number of chromosomes will be restored. Then you'll have determination of the embryonic sex. That is, you can be able to know if it's XX or XY genotype. So by studying the zygote at this time, you can be able to tell whether the embryonic sex is either male or female. And of course, due to fertilization, various variation of species will happen due to genetic variation. So genetic variation is a product of fertilization. Then, of course, you'll have metabolic activation and restoration of capacity for cell division. So remember, you'll have the diploid nature being restored, and due to that, you'll have metabolic activation and restoration of capacity for cell division. So that cell division will be known as cleavage. So we can start with implantation. So remember, implantation is attachment and burrowing of the endometrium. So remember, the endometrium is the innermost layer of the uterus. The uterus has three layers: endometrium, myometrium, and perimetrium. So attachment and burrowing of the endometrium on the blastocyst into the endometrium is known as implantation. So it normally happens in the fundus of the uterus. So it happens on the fundus. So this is the fundus, on the posterior aspect of the fundus. That's where it normally happens. Though sometimes you can have abnormal implantation, and that is what is being shown in the images here. So you can have abnormal implantation, which happens in the ovary, giving you ovarian implantation. It can implant in the infundibulum, giving you the infundibular implantation, in the ampulla, giving you tubal implantation, the isthmus giving you tubal implantation. It can happen on the fundus, but now on the border between the isthmus and the fundus, it will give you interstitial implantation. It can also happen in the abdomen or on the outer wall of the uterus, which is the peritoneal implantation. It can also happen in the cervix, which is cervical implantation, or now abdominal implantation. So ideally, there is no better implantation that can happen apart from the one that happens in the fundal implantation. So this one will lead to ectopic pregnancy. So ectopic pregnancy, which will happen in those parts. So kindly take note of those parts that I talked about: ovarian pregnancy, abdominal pregnancy, cervical, peritoneal, infundibular, tubal, and interstitial will lead to ectopic pregnancy. So you need to discuss the normal karyotypes. So the normal karyotypes in females will be 46XX. So you can count these from 1 up to 22. So up to there you have 22 pairs of chromosomes. And then you'll have these sex chromosomes, which of course, if it's female, you'll expect to have two X chromosome, absence of a Y chromosome. So that will be female karyotype 46XX. In males, you'll have 22 pairs as in females, but now in the sex chromosomes, you'll have one X and one Y, giving you 46XY, which is a normal male karyotype. So remember, you can have genetic or chromosomal abnormalities that you can see here, which are very common in exams. So for example, Down syndrome, which is trisomy 21, will have an extra chromosome in chromosome number 21, in a pair of chromosomes number 21. Edwards syndrome, which is trisomy 18, of course, tri means 3, extra chromosome in 18. Patau syndrome, trisomy 13, extra chromosome in 13. Turner syndrome, which is monosomy X, ideally you'll have single X chromosome in females. Klinefelter syndrome, 2X chromosomes in male, so that is now XXY. Then triple X syndrome or the super females, you'll have 3X chromosomes in females. Then XYY syndrome, which is 2Y chromosomes in males. So this one is a rare abnormality, but it's present. So some of the chromosomal abnormalities that you can have. So it is important that you know this and to even appreciate how the karyotypes of themselves of these diseases, of these abnormalities are usually there. So we'll go to the first week of development. For this first week of development, you have day 0 being fertilization. Day 1, you'll have the 2-cell stage. Then day 2, the 4-cell stage. Then day 4, you'll have the embryo. Day 3, you'll have the embryo. Day 4, you'll have the morula. And then you'll have blastocyst. So this time you'll have this early blastocyst and then the late blastocyst, which happens during day 6. So implantation happens around day 6, day 7, as you can see here. So the stages of, you can see now, fertilization day 0, then it start to move. Remember, this is only happening in the ampulla. So as it moves, the process of cleavage, hatching will be happening. And this one will be covered specifically in a video on the first week of development, so don't worry. So just note that fertilization day 0, then have 2-cell stage, day 1, 4-cell stage, day 2, the embryo day 3, modular day 4, then you'll have the blastocyst, which is happening in, you can have early blastocyst, that is before hatching of the zona pellucida, and then late blastocyst, that is after hatching, removing the zona pellucida. So note there is increase in number of cells. So cleavage is happening, mitosis is happening a lot. So you go to the second week of development. So second week of development is commonly known as week of twos because you'll have 2-cell masses, that is epiblast and hypoblast. 2 poles, embryonic pole and abembryonic pole. 2 embryonic layers, trophoblast, cytotrophoblast and cytotrophoblast. 2 cavities, yolk sac and amniotic sac. 2 trophoblastic layers that we'll see in a few minutes. So this is what happens from day 6. So remember, day 6 and day 7 is the first week of development, and that's when implantation is happening. So from now day 9 to day 11, you can appreciate implantation has already happened, and now the week of twos is at play. So when you're talking about the week of twos, you'll have 2-cell masses, as you can see here. So this is the epiblast, and then you have the hypoblast, 2-cell layers. Then you have 2 poles. One pole is here, embryonic pole, and then the other layer will be the embryonic pole. The other part that we need to appreciate is 2 embryonic layers. You can appreciate here you have cytotrophoblast, and then in green is syncytiotrophoblast. So 2 layers, 2 embryonic layers. 2 cavities, amniotic cavity, and the primitive yolk sac or the exocoelenic cavity. Then 2 trophoblastic layer that we've talked about, syncytotrophoblast and cytotrophoblast. So it is important at least we can be able to appreciate the week of twos in terms of those layers. So by the end of the second week of implantation, that is what is happening. You have 2-cell layers and everything. So what will now make us to know that you are moving to the third week of development? So when you have now this bilaminar disc, the 2-cell layers becoming 3 layers. So from 2 layers of these cell masses, will have 3 layers. And before then now, let's have a summary of this. You'll have fertilization on day 1. or day zero actually. Cleavage is happening. Compaction due to cleavage, mitosis is have compaction. Then differentiation of the cells, cavitation, that is development of the blastocoelemic cavity needs. It is the process of hatching from the zona pellucida, implantation on the endometrium, then cell mass differentiation, that is, you have epiblast, hypoblast, and trophoblast. Bilaminar disc formation, that is now the epiblast and the hypoblast being formed. Then you have the process of gastrulation happening in the 12th up to day 18. Then amniotic sac enlargement. And that is now will lead us to the third week of development, which is gastrulation. And in this third week of gastrulation, you'll have the process of formation of three germ layers. So gastrulation is the process of formation of three germ layers. The three germ layers will be ectoderm, mesoderm, and endoderm. It's important to note that these three layers, all of them will be coming from the epiblast. So how? You have epiblast and hypoblast. In between, you'll have formation of a space. So in between, at the center of the epiblast, you'll have formation of a primitive streak. It is important, first of all, you know the function of this primitive streak. So primitive streak, as you can see, this is a primitive streak, and actually it will be there in spot images in exams. So you can appreciate this is a primitive streak. We have the primitive node here and the primitive bit. So what happens is that this primitive streak, it will act like an avenue where cells of the epiblast will lose their characteristics and migrate, as we'll see. So what the function of the primitive streak? It provides, first of all, structural support to the bilaminar embryonic disc. Remember, at this point, when you're developing the primitive streak on the epiblast, you have two layers, epiblast and hypoblast. So structural support to the bilaminar disc. Then it establishes on the embryonic axis and bilateral symmetry. Remember, it will develop at the central position of the epiblast of this bilaminar disc. So it is established the embryonic axis. So this one will be left, this one will be right, and the bilateral symmetry. So no side, no part is larger than the other one. And then is the one that will direct the process of gastrulation, process of formation of the three germ layers or the trilaminar embryonic disc. And what is the fate of this primitive streak? It's supposed to degenerate. If it doesn't degenerate, you'll have anomalies. So that is the primitive streak. So primitive streak will lead to this. You can see this is a primitive streak. So cells will come towards it, have that migratory property, and then they will come and displace the hypoblast in the first migration. In the second migration, they will fill up this space in between. And due to that, you'll have the three layers. So the three layers will have the remaining cells becoming the ectoderm, the cells that fill up this space being the mesoderm, and the cells that displace the hypoblast to become the endoderm. But we'll see that well in our next videos. But in before, we need to talk about this process of gastrulation. So the process of gastrulation happens in that you'll have the epiblast. This is the epiblast. The cells really come towards the primitive streak. They get that migratory property, that is anterior posterior polarity. And then first of all, they displace the endoderm. They displace the endoderm. They displace the hypoblast to become the endoderm. You can see these cells are actually different in terms of color coding, showing this is a first migration phase, first phase of migration. Then the second phase of migration, they will come and the cells of the epiblast will come and fill the space in between the epiblast and the developing endoderm to give us now the mesoderm. So from there now, this is what you'll have. You talked about this. This is a primitive streak. This is now the epiblast and the hypoblast. These are the cells migrating. You can see first of all the first phase giving us the endoderm. The second phase giving us the mesoderm. So what will be the derivatives of gastrulation? But before then, maybe we can look at what are the derivatives of the process of. Yeah, and now you have derivatives of the process of gastrulation, and that is what we can be able to see here. So on the first image that I would like us to see is actually what happens when you have the derivatives of when you have gastrulation. So remember you'll have the outermost layer being the ectoderm, the middle layer being the mesoderm, and then you'll have the inner layer being the endoderm. So that is what we are having after gastrulation. But before then, the mesoderm will undergo some differentiation into three layers. So we have the notochord at the center, which will direct the process of formation of the nervous system, known as that is neurulation. So the notochord, the one this black dot here, of course now it will lead the process of neurulation. So the mesoderm that will be just next to it will be known as paraxial mesoderm, that is next to the notochord. And then you'll have in between the paraxial mesoderm and the outer part, we'll have the intermediate mesoderm. Then on the outer part, we'll have the lateral plate mesoderm, which will be divided into two. And you can see here now, you'll have the process of neurulation being directed by notochord to give us the neural tube. The neural tube will give us the nervous. And actually, you can see it's coming from the ectoderm. So one of the derivatives of ectoderm will be the nervous system, but we'll talk about in a few minutes. Then by the end of it, only you can see you have the paraxial mesoderm, the intermediate mesoderm, and even now the lateral plate mesoderm being well differentiated here. Yeah, so this just to show us the derivatives of the three germ layers that we talk about. So finally, this is what we'll have. We'll have the ectoderm, the one in blue. So specifically, that one will be surface ectoderm. Then this will be neurotube coming from the neuroectoderm. This is a neurotube. We'll have the notochord. These two are actually developing aorta. This is a paraxial mesoderm, intermediate mesoderm, and then these are the two parts of the lateral plate mesoderm. So this one will have the somatic part of the lateral plate mesoderm. This will be the splanchnic part of the lateral plate mesoderm. And not forget that here we have the endoderm. Yes. So let's talk about the ectoderm. So for the ectoderm, it is being referred to as the protecting and communicating layer. Protecting, why? Because it's the one that will give us the skin, the integumentary skin. And then communicating because it's the one that will give us the nervous system. So the derivatives now will be neuroectoderm. So this is the neuroectoderm that will become the nervous system. This is well described in our video to do with neurulation and development of the nervous system. So if you are keen enough, if you like to learn more about this, you can go and visit our video on neurulation. Then surface ectoderm. So surface ectoderm, this one will become the skin, be given that is on the outer part, so the protective layer. So it will form the epidermis of the skin. So if you look at skin, that's actually a product of surface ectoderm embryologically. You can go to now the mesoderm. So remember the mesoderm we talked about has three parts. We have this being the paraxial mesoderm, the second part here being the intermediate mesoderm, and then the third part here being the lateral plate mesoderm. So we started off by talking about paraxial mesoderm. So this paraxial mesoderm. So you can just figure out just next to the nervous system, developing nervous system. So it tells you, it will give you the bones of the axial skeleton in addition to other things. So undergoes the process of segmentation to form somite. So this one isn't actually a developing somite here. And the somite will divide into it will give rise to three parts. It will give rise to sclerotom, myotome, and the dermatome. But before then, somite first of all divides into two: sclerotom and the myotome. So sclerotom gives us exoskeleton. Then the dermatome part of myotome gives us the truncal dermis, that is the dermis of the back. And then the myotome part of myotome gives us the skeletal muscles, specifically of the trunk. So somite or the paraxial mesoderm will give rise to exoskeleton, will give rise to skeletal muscles of the trunk and the truncal dermis. So remember, the skin has epidermis, dermis. So epidermis coming from the ectoderm, and then the dermis specifically of the back will be coming from the paraxial mesoderm. So those are the derivatives of the paraxial mesoderm, which is a part of mesoderm. The three parts. Kindly take note of those derivatives, especially where they come from. Sclerotom gives us exoskeleton, myotome, skeletal muscles, dermatome gives us truncal dermis. Now we go to the intermediate mesoderm. So in between here, we'll have the intermediate mesoderm between paraxial mesoderm and the lateral plate mesoderm. So it's called intermediate mesoderm because it's in between the two. So between paraxial mesoderm and the lateral plate mesoderm, what are the derivatives? You'll have two systems coming from it. You'll have the urinary system. Talk about urinary system, talk about the bladder, the ureter, the kidneys will be coming from the intermediate mesoderm. Then we'll have the reproductive system, especially the internal genitalia for both males and females. Will be so now talk about the ovary, the vas deferens, the fallopian tube. All of them will be coming from this intermediate mesoderm. So the reproductive system of both males and females, that is internal genitalia specifically, will be coming from intermediate mesoderm. So the two processes, the two systems, urinary system and the reproductive system, will be coming from intermediate mesoderm. And now we go to the lateral plate mesoderm. So remember we talked about the lateral plate mesoderm being divided into two. The somatic, the one that now is actually on the outer bit, that is the somatic or the parietal part of lateral plate mesoderm. And then you have the splanchnic or the visceral part of lateral plate mesoderm. So it's important that you note that lateral plate mesoderm will be divided into two: splanchnic or visceral part, parietal or the somatic part. So the other the derivatives of the splanchnic. As you can see, this splanchnic will be incorporated together with endoderm when you have the lateral folding. So remember, you'll have this yolk sac that you can see here, and then this amniotic cavity. So what happens is that some part into this developing embryo once you have the lateral folding. So that means the endoderm is some part of the splanchnic part of lateral plate incorporated into this developing embryo. And so due to that, the splanchnic part of lateral plate mesoderm will give smooth muscles of the gastrointestinal system and the bladder, and other smooth muscles of the body will be coming from the splanchnic part of the lateral plate mesoderm. It will also give us the cardiac muscles of the heart. Then it will also give us the visceral connective tissue. So sorry for that. The visceral connective tissue will be coming from the splanchnic part of mesoderm. So the three derivatives: smooth muscles of GIT and bladder, cardiac muscles, and the visceral connective tissue. Then the derivatives now of the parietal part of lateral peak mesoderm will be the appendicular skeleton, the dermis. So remember the dermis of the other parts of the body, apart from the back, will be coming from the parietal part of lateral peak mesoderm. So those two appendicular skeletons will talk about the bones of the arm, the bones of the forearm, the bones of the upper limb and lower limb. They will be coming from the parietal part of the lateral peak mesoderm, and the dermis will be coming from that. Okay, so we go to the last bit to do with the endoderm now. So you talked about the ectoderm giving us the skin, that is a surface ectoderm, integumentary system, and the neuroectoderm giving us the neurotube, which gives us the nervous system. We talked about the mesoderm being divided into three: paraxial mesoderm, intermediate mesoderm, and lateral peak mesoderm. So paraxial mesoderm we talked about as giving us the axoskeleton, the truncal muscles, and even some muscles of the appendages. And we talk about now even the truncal dermis. Intermediate mesoderm giving us the reproductive system and the urinary system. We talked about the splanchnic mesoderm giving us the smooth muscles, cardiac muscles, and the connective tissue of the various organs, various viscera. We go to lateral plate mesoderm, the parietal part, or the somatic part, which give us the appendicular skeleton and the dermis of the remaining part of the body. So what is remaining is just to talk about this endoderm, the one in yellow. So this endoderm, remember, it will be incorporated during the folding, and due to that, it will be the nourishing layer due to its derivatives. And the derivatives will be three derivatives: the epithelial lining of GIT. So remember, we have the connective tissue coming from the splanchnic part. So the epithelial lining of GIT will be coming from endoderm. Then the parenchyma of the glands of the digestive system. For example, the parotid gland, the liver. So the cells, the parenchymal cells, will be coming from the endoderm. So the pancreas, the parotid glands, mandibular glands, liver, the parenchymal cells will be coming from the endoderm. And then the epithelial lining of the respiratory system also will be coming from this endoderm. Remember the viscera, the connective tissue of the respiratory system will be coming from the splanchnic mesoderm. So remember the visceral connective tissue of all the organs will be coming from visceral part of lateral plate mesoderm. Then the epithelial lining of the respiratory system and also the epithelial lining of GIT will be coming from the endoderm. Then the parenchyma of the glands of the digestive system will be coming from specifically the endoderm also. So that's majorly about general embryology or the basic embryology. So we also have videos that will be covering systemic embryology, talk about videos on neurulation and which will lead to formation of the nervous system. A very interesting video. Feel free to watch it. Development of limbs, a very good video with a good number of views. It's actually very simple, about 20 minutes, direct to the point. Feel free to watch the video. Development of eye and ears, very good videos here, but now this one will cover it systematically. Development of head and neck structures, how they develop from the various brachial apparatus or the phalangeal apparatus. Development of the exoskeleton from the paraxial mesoderm. Development of the intermediary skin or the system from the ectoderm. development of the urinary and reproductive system, development of the digestive system and the related glands, and all these actually we've covered it. We've covered it in our video. So feel free to watch the videos, and that will be systemic embryology. Remember, this was just introduction to embryology, so giving us what embryology is all about. Yeah, and so with that, I just wish you all the best in your career. Stay focused and never give up. Always study hard and hard. Remember, embryology is one of the simplest part of anatomy. And whatever you do it wholeheartedly, doing that, like you're doing it for yourself, for the betterment of education. So that verse, Colossians 4:23-24, I wish you all the best in your academics. Feel free to like, comment, share, and subscribe for more videos. And even from myself to you, I just wish you all the best in your career, all the best in your education, and I have nothing but thank you. All the best.